Hey, well, welcome everyone. Thank you all for joining us. Have yet another uh, awesome URIG group of speakers today. We have two speakers. Our first speaker of the day is going to be Dr. Katy Patras, who is assistant professor in the Department of Molecular Virology and Microbiology at Baylor College of Medicine in Houston. And and then we also have a second speaker today, Seth Reisner, who's an MD PhD student in Maria Hadji-Francescu's lab at Vanderbilt. And I know both of these speakers, and I've followed their science for a while now, and I can tell you that they're doing really awesome things. And I'm super happy that we have the opportunity for them to share their science with our community. So, Dr. Petras, I'll go ahead and give it over to you, and we can get going.
All right, just quickly double checking. You guys can see my slides. You guys can hear me, so I'm not talking
Yeah.
into the void. Okay, perfect. So I'd just like to thank Mike and Mariana for organizing this, Mike for the invitation to join this group. My work is tangential, so I'm going to say vaginal microbiota more than I say urinary microbiota. But I do have one slide. But thank you for the invite. And I'm going to focus today on one of the main research areas of my lab, which is trying to understand how uropathogen colonization of the vaginal tract is a key risk factor for infectious disease, including UTI. Okay, so I know we all know this, but to do my due diligence, we study urinary tract infections. This is one of the most common bacterial infections worldwide, about 400 million cases globally each year and almost 250,000 deaths. You can see here from this chart, it's something that's increasing in prevalence. I know it's a little bit difficult to read. Here, male incidence is in green, female incidence is in this reddish pink color. And you can see very quickly, this is primarily a disease that impacts females. One of the urgent needs related to UTI treatment is this antibiotic resistance crisis. UTIs are one of the leading reasons that we give antibiotic prescriptions. And you can see the sheer abundance. This together is driving AMR and these uropathogens. And so there's this urgent need for alternatives to antibiotics. And this is just another graph, same paper. Now, instead of plotting it over chronologic time, it's showing it over the lifespan of an individual. And you can really see here that women of reproductive age bear the brunt of urinary tract infections. And so that's primarily the population that we're trying to study and develop therapies for in my lab. Okay, so why are women more susceptible to UTI? I am not answering this question today remotely. I know this group is interested in this question, but I don't have an answer for you. But just highlighting some of the theories that have been established over the years. Certainly, there are recognizable anatomical differences between male and female. Perhaps that plays a role. I think we've kind of debunked this idea, but hygiene was thrown around for a while as perhaps the reason why females might be more susceptible. And then very often what happens when there's a sex difference and we don't know what to do, we kind of shrug and say hormones. So probably hormones are involved, but that's not a very mechanistic answer. Okay, so how my lab thinks about this problem is trying to understand a little bit more. Okay, well, can we understand where these urinary pathogens are coming from? And so this is another pie chart that I know this group is familiar with, very widely cited review that's looking at the agents that tend to cause uncomplicated UTI. So when there's no underlying comorbidities. And so by far, the biggest chunk of the pie is this uropathogenic E. coli, which is what I'm going to be talking about today. But you can see from this list, there are multiple other bacterial and fungal species represented here. And a common theme that we can see through all of them is these are organisms that colonize our various microbiomes. These are present throughout our body, not necessarily associated with disease, but certainly contain the capacity to cause pathogenesis. And so there's, of course, some of the areas that these organisms tend to colonize are areas that we have established microbiota. So the gut is a widely accepted source or reservoir for uropathogens. This group is quite aware that there's also a urinary reservoir potentially for some of these organisms that tend to cause recurrent urinary tract infection. But what's missing on this slide is what I'm going to talk about. So the vaginal microbiota, certainly something that's unique to females and something that could also be an additional reservoir for uropathogens. Here's my one slide. I know this group is familiar with this as well, but here's a study from Dr. Wolf's group looking at the similarities of the bladder microbiome with other related microbiomes. And you can see there's much more overlap with the vaginal microbiota, suggesting a lot more shared organisms versus the GI. And then in this really nice study, they actually sequenced these isolates both from the bladder and the vagina and saw 99 point whatever percent identity, suggesting that they truly are the same clone that's colonizing both of these sites. Okay, so let's now switch to talking about the vaginal microbiota for the rest of this talk. So again, I'm sure this group is aware, but just to bring everybody up to speed, the vaginal microbiota in women is quite unique compared to what we typically think of when we think of microbiomes. So tends to have a very low alpha diversity or low complexity, tends to be dominated by a single species, predominantly lactobacillus. As you can see here, I've highlighted some of them. So crispatus and inners tend to be the two dominant taxa, but there's certainly several other lactobacilli that dominate. But what you can also see is some of those, potential urinary pathogens that we saw exist here. So there's streptococcus, there's staphylococcus, there's enterococcus. They don't tend to be dominant, though occasionally you'll see a vaginal microbiota that is dominant. They tend to be, more rare and in lower abundance. But I do want to highlight there's this community state type composition of the vaginal microbiome that instead of having a dominant lactobacillus has a mixture of other different anaerobic organisms. And it's this group that tends to be at increased risk for urogenital infections, including UTI, but also other infections as well. Okay, so just to give you some additional evidence of why we think the vaginal microbiota is a source for uropathogens comes from some of these papers. So here is a study that looked at vaginal E. coli colonization in women that either had recurrent urinary tract infection or controls, and they were saying whether or not E. coli was there. And you can see that there's a much higher prevalence of vaginal E. coli colonization in women that have recurrent UTI, so closer to 40% versus controls that are closer to 10 or 15%. There's also been some more descriptive studies that have looked at, does it matter the other organisms that are there in the vaginal microbiota? And so here's some data suggesting that when lactobacillus are not present in the vaginal microbiota, there's an increased rate of E. coli vaginal colonization versus when lactobacillus is present. There's still some, it's not completely exclusionary, but the rate of colonization tends to be related to lactobacillus presence. This is another study talking about bacterial vaginosis. And in this cohort, these women were symptomatic, so actually showing signs of dysbiosis in the vaginal tract. They saw that in women, particularly non-pregnant women that had bacterial vaginosis, they had an odds ratio of 50 more likely to be UTI positive than those that did not have BV. So definitely strong clinical correlation for lactobacillus dominance being associated with the absence of E. coli in the vaginal tract. Okay, so that leads you to the very logical question, if you weren't already there. Vaginal lactobacillus, do they directly inhibit E. coli? Is that the reason why we see this inverse correlation? There is quite a bit of data out there, particularly in vitro, suggesting that there, of course, is anti-E. coli activity in vaginal lactobacillus. I don't have time to show all of that today. I'll just show you what we do. And so my lab is very interested in understanding, are there particular characteristics of certain isolates of the vaginal tract that are protective? Why are 80% of women not colonized with E. coli and why are 20% colonized? Is there something that's protective in regards to a vaginal species that would be there? So we have healthy human vaginal swab samples that we collect single clonal isolates of bacteria. We mix them 1-to-1 with our uropathogenic E. coli isolates. We select a media that's very similar to the vaginal tract, so it's a simulated vaginal fluid. We also do these screening in a microoxic environment to mimic more of what they would see in a human vaginal tract. So we mix these vaginal isolates with our E. coli, 24 hours static culture, and then plate and look and see, is there any change in the recovery of E. coli? And so here I'm showing you a typical result of this screen. The different isolates are colored by the species that they are. But you can see, we see quite a range of inhibition. So our baseline E. coli controls would be at this 100%. We see some strains that inhibit down to 1,000 folds or more, and then some isolates that do absolutely nothing to affect E. coli growth. And you can see it's not a specific species that's doing this, it's very strain dependent. So this is absolutely fascinating. So is there any evidence that E. coli that can inhibit vaginal isolates that can inhibit E. coli, do they have any actual protective activity in preventing against UTI? So there are a couple studies. This is one from Ann Stapleton testing a Lactobacillus crispatus vaginal isolate that's being developed as a biotherapeutic candidate. It's called Lactin-V. And this was a study that was pretty decently powered. They took women that had acute UTI, treated them for UTI, and then put them in the placebo group or the Lactin-V cohort. They gave Lactin-V as a vaginal suppository for a week, and then they tracked whether or not they developed a subsequent UTI in the following period. And so what you can see here, when they just straight compared Lactin-V versus placebo, there was no difference in relative risk, so no significant clinical impact. But when they looked within the Lactin-V cohort via 16S PCR at the levels of Lactobacillus crispatus, so not necessarily Lactin-V, but just total Lactobacillus crispatus, that's where they saw a significant effect in women that had higher levels, defined, I think it was like 1 million copies of lactobacillus, were protected against recurrent UTI versus the group that had lower levels of lactobacillus. So again, very suggestive that there is some protective activity that we could capitalize on. But we're far from optimized. And so this is certainly something that my lab is very interested in studying. The problem is we either need to do massive amounts of clinical trials with all these different isolates that everybody is collecting everywhere, or we need better preclinical models to predict what biotherapeutics are going to be effective in the clinic. And so we picked option B. And I'm going to tell you a little bit about some of those models. So the first has been to really try to understand, how do we model the vaginal microbiota of humans in vitro? It's not as complex as the gastrointestinal tract, of course, but there still are multiple species. It's still something where we can see the ecology of interactions between pathogens and commensal microbes. And so we spent a lot of time trying to develop some of these models. We very conveniently, my lab is neighboring with a lab that studies the gastrointestinal tract. They developed this continuous flow bioreactor system for gut bacteria. And so we've adapted that to study vaginal microbes. Here's schematic here. You know, kind of hard to convert these dimensions. What do these mean? These hold about 10 mils worth of media. We have about 6 in these 3D printed strips, and they're continuous flow systems. So there's constantly new media being introduced, waste media being removed, and then there's a sample port that also lets us collect samples or add things. So this is the basic model. It's pretty small, which is why it's called a mini bioreactor. This is, you know, to scale someone's hands. We have pumps flowing new media in, waste pumping media out, and then they're on a stir bar where we can agitate the media in the reactors. I think these are Claire's hands. I've had 2 graduate students put a lot of time and energy into this model, and so they're highlighted here. But the system is small enough we can actually put it in its entirety into an anaerobic chamber or a microoxic chamber, and so that's one of the main selling points there. Okay, so what does this reactor model? What does our data look like? So we've taken healthy human vaginal donors, swab samples. We make glycerol stocks. We also inoculate them straight into these bioreactors. As I mentioned, we use a simulated vaginal fluid that is adapted from a prior protocol that models things like the nutrient sources, the pH, buffering capacity. We also do this in a microoxic environment. We have a pretty low retention time, basically as low as we can get away with without clogging everything. And this is to model, again, the turnover of fluid produced at the vaginal mucosa. And so here I have, I think it's 6 different donors that are grouped into different community state types. So you can kind of see the first 3 community state types are dominated by lactobacillus. CST4 is a non-lactobacillus dominant community state type. We're able to grow decently high cultures of bacteria from all of these different samples. And then interestingly, we also see acidification of the media to levels that we'd expect of the vaginal tract, but it's very donor community dependent, which is really fascinating to us as well. So some acidify to a greater degree than others, even though there's robust growth in all of them. We've also looked at the composition of these communities by collecting samples and then doing 16S analyses. So here I'm showing you a PCA plot of 3 different representative communities. So the darkest circle, that's actually the 16S composition of the input swab. So what we sequenced on the swab that we collected. And then the lighter symbols are from replicates of the bioreactors collected at day 4. And you can see they cluster very similarly to the input sample, with perhaps a little bit of an exception of community state type 4. You can see some of the taxa that are driving this variability there as well. So certainly something that's not perfected, but definitely is modeling some of what we're getting on our input swabs. All right, the other aspect that we need to model is the host. So how do we understand how the host epithelium responds? How can we test therapeutics when there's a host component? And so this has been work that's been driven by, now you can see we're in logarithmic phase. There's even more students here. This has been a massive effort. This has been in collaboration with Dr. Sarah Blue and the organoid core here at Baylor. We've generated human vaginal organoids, and these are actually non-transformed stem cells that we collect from healthy donors via vaginal scrapings. We actually use a cervical brush, the same that was used for pap smears, to collect these epithelial stem cells. We propagate them in matrigel to generate these spheroids. We add a massive amount of growth factors to achieve this. And then once we have sufficient density of these cells, we actually dissociate the spheroids and plate them onto transvolts. And here's where we actually differentiate them into an air-liquid interface. So we actually have air on the apical side, media on the bottom to get that final differentiation. So some of the data that we've gotten from these organoids that suggest that they are actually modeling the vaginal epithelium, we found that they've produced high levels of protein that we'd expect for a keratinized epithelium, including keratins and mucins. Here's an H&E of the differentiated layer. It's a stratified squamous epithelium. And then when we've done bulk RNA sequencing, we've paired this to human reference data from entire tissues. Here I'm showing you data. These are 4 different donor lines of organoids clustered with human vaginal reference RNA-seq data, and then they cluster separately from some of these other organs, including other organs within the human reproductive tract. So again, we think that they're pretty distinctly modeling the vaginal tract. Okay, so what do we do with these? Well, many different things. In the context of this talk, I'll talk you through, some of the things that we're using to develop some of these biotherapeutic candidates. So we take our differentiated organoid cell layers, we add vaginal isolates from those healthy donors first, and then we add uropathogenic E. coli in this example, kind of to model pathogen invasion of a community. And then we collect all these different types of samples. We look at cytotoxicity, we look at cytokine production, and then we look at what happens in terms of bacterial dynamics on the epithelium. So just to show you a little bit of data of what we tend to get from these organoids. Here, each individual point is 2 independent replicates from the same donor line, which are very consistent with each other. But you can see the sheer spread of donor to donor cell line, which is actually really fascinating to us. I'll bring that up one more time later. But here we're looking at how these vaginal isolates impact E. coli adherence to the vaginal epithelium. You can see, even within the same species, we see some strains perform very well at reducing adherence, whereas others do not do anything. So very, very fascinating to us, and might explain a little bit of why, across community state types, we can see E. coli colonization, even though, in vitro, sometimes we see inhibition. So there's a lot to go through here. So what we're doing as in terms of development is to try to take some of the key properties that we get from our screening of our individual isolates in both in vitro and on these organoids to try to develop synthetic communities that actually accomplish a collective function. And so, so far we've started with fairly simple synthetic communities. We picked 3 because 3 is a great number. That's the rationale. 3 sounds feasible. But we've picked them based on some different parameters. So synthetic community 1 of the data I'm showing you is selected based on its ability to inhibit pathogens. There's a synthetic community 2 that's primarily geared towards host modulation, so an epithelial response or an immune response from the epithelium, and then a synthetic community that has both of these features. So the isolates that assembled that ZenCom can do both of these. So we assume that synthetic community 3 is probably the most ideal approach, but we just wanted to cover our bases. Do these all look different, or do they all look the same, or is one better than the other? So here's some representative data from MBRAs, those continuous flow bioreactors. This is upon multiple challenges with uropathogenic E. coli. So what we do is we first assemble the communities in the bioreactors and let them stabilize for several days. And then we challenge them with 2 different doses of E. coli a day apart. So here are the 3 synthetic communities that I mentioned have these properties. We also assembled a non-inhibitory ZenCom to represent bacteria that do not contain these properties. And then this clear circles our UPEC strain alone. And what you can see is that within 4 hours of the first challenge, these communities are really effective in clearing the pathogen versus the inactive community or the E. coli only community. When we challenge again, we see a little bit more of a disturbance, but we see some resolution again with known detected E. coli within 48 hours. We're seeing some interesting dynamics with the non-inhibitory ZenCom. I have some ideas on this. I mean, we're actually running an evolution experiment, if you think about it. So I think there's some interesting adaptations that are occurring, but we're still interested in exploring that some more. What we've done with these synthetic communities on the vaginal organoids, we've looked at cytotoxicity, we've looked at cytokine production. What you can see here is that the communities with protective effects or beneficial effects seem to be protective in reducing cytotoxicity in response to pathogenesis compared to the inactive community. We're starting to see some evidence for the immune modulation in our host-targeted communities. We're still building the end there, so this is very preliminary, but again, they're starting to separate in a very interesting way. Okay, so I just have one more tangential section, but I just wanted to kind of recap where we are here. We are building evidence that vaginal lactobacillus could contain some beneficial properties to actually control uropathogen colonization of the vaginal tract, which we believe will be a key feature in preventing urinary tract infections. Okay, but vaginal bacteria are not the only thing there, most likely. They're probably not the only thing that this group is interested in. There's definitely an obvious other biotherapeutic agent that came to mind when we were designing these. Maybe you guys have thought of it. Bacteriophage are there in pretty high abundance. And so that's certainly an area that was very logical, tangential story to start. So I'm just going to show you a little bit of data that we have with bacteriophage. So this is work that's been done by a postdoc in the lab, Vishnu Joshi. This is using a anti-UPEC targeting phage HP3, which our collaborators Anthony Moresso has worked on in a variety of different pathogenic E. coli types. And we started, just in basic asking, can bacteriophage reduce vaginal colonization of E. coli in vitro? And so we wanted to model kind of these 2 different states. So one is, phage everywhere. There's phage in the supernate and there's a phage adherent to the surface. This would be, kind of what would be modeling active phage therapy. And then we also had a state which was, where we only allowed adherent phage to be present in that community to kind of model what we'd maybe expect under more endogenous settings or, kind of post phage therapy. And then we did a pretty standard E. coli adherence assay, 30 minutes. And then quantified both PFU for phage and CFU for bacteria. So just to show you the phage data first, we quantified, 3 orders of magnitude higher of phage when phage are also contained in the supernatant. But interestingly, the cell-associated phage were the same in both conditions. So really, there seems to be kind of this prime maximal real estate of the phage interacting with the host epithelium. And this is all in the absence of bacteria. So there's something there where the phage are actually able to bind. We don't know what they're binding, but they appear, at least a fraction of them, to stick around. When we look at the bacterial levels compared to a mock control, HP3, that's the single phage that we've primarily been focused on, but we've also tested a phage cocktail. This is a 3-phage cocktail that contains HP3, but several other phages that have also shown additional anti-UPEC properties. The hope is that, we think as a field, phage cocktails might be beneficial because you could have multiple bacterial targets that are being targeted, host immune neutralization may be different. And so we think cocktails might be the way to go, but we got to generate data to show that. Our data here is certainly showing that the cocktail is showing the most efficacy in terms of removing E. coli from the vaginal epithelium, though we do see some effects of the single phage as well. There's a lot going on here. We have host cells, we have phage, we have bacteria. And so we've also started to visualize the dynamics of these three using microscopy. And so this is a collaboration with another postdoc in an adjacent lab as well. So here we're labeling host nuclei as well as we turn agglutinin to label the host membrane. We have an RFP containing uropathogenic E. coli strains. So you can see here in our UPEC only controls, there's these beautiful red bacteria. And when we add phage, we actually found a way to label phage using another nucleic acid dye, a cyber gold, and that actually binds the phage nucleic acid. And it's bright enough that we can actually see phage, which are very small. And so here you can kind of see them in green. We don't know whether or not these are intracellular or extracellular. There's definitely some reports that phage can be taken by host cells. We don't yet know if that's the case, but you can see how much fewer bacteria there are when phage is present. So it's certainly doing something. We think we're seeing occasional co-localization of phage and E. coli too. So we're really wanting to tease apart, where are the phage, where's the bacterium, where is killing occurring, and hopefully this microscopy is a way to evaluate that. Okay, microscopy is great, but it's not near as complicated as mouse models, which is, kind of the next preclinical step for phage development. And so we have adapted a mouse E. coli vaginal colonization model that's widely used in the fields. We synchronize them with estrus to get them all in the same estrus stage, give them a single dose of vaginal E. coli, allow that colonization to take place for 24 hours. And then in these experiments, we actually dosed with phage every day for 7 days and then collected tissues at the later time point. And just before we inoculated the phage dose, we'd take a vaginal swab to quantify bacterial burdens as well. Okay, so here's the results. They're a little underwhelming, but that's okay. We're all about optimizing some more. So here in this first graph, we're showing basically percent colonization. So when the mice actually clear E. coli below the limit of detection, you can see that actually the vast majority of mice remain colonized throughout this experiment. We do get statistical significance, but again, we're talking about 90% versus 97%. So it's modest, but there is an effect. Here I'm showing you the vaginal CFU. Again, you can appreciate the sheer variability. It's all very nice and clustered at the beginning, and then things just kind of get pretty spread out. We did get statistical significance on one day with the phage treated group being lower, but again, likely not that impactful. So we're really interested in continuing to build, how do phage cocktails come into play? Is there a host reservoir that the bacteria is hiding out in that the phage can't penetrate? Are the phage being neutralized by a host factor? So these are things that we're interested in pursuing. Okay, so really quick, I just wanted to, hopefully tell you what I told you, and then we can go into some questions. So, we think uropathogen colonization of the vaginal tract is the key risk factor for UTI. We need to develop therapies to control colonization. What is the ideal candidate? We don't know. We have some ideas. Happy to discuss this with you further. These mini bioreactor arrays that we have developed can model a vaginal community in its entirety. It's a very useful tool for screening biotherapeutic candidates. what's going on in these reactors, how are the microbes interacting with each other, what kind of nutrients are being played on, do we see any synergistic activity of different microbes? This, could go on many ideas about this different tool. And then finally, human vaginal organoids appear to model the human vaginal epithelium and also serve as a useful tool to screen. One of our big questions is, what is the role of host genetics? Why do we see such a drastic effect on our different donor lines? One thing that we have done as a part of the study is when we're collecting samples for the vaginal organoids, we're also taking the microbial samples from the same patient. We haven't done it yet, but what I'm really keen on doing is pairing together the microbes and the donor line of the organoid and really seeing, how collectively do they assemble, is there host specificity in that interaction? So really interesting future direction. And with that, just want to thank everybody in my lab. They are amazing. The people here in bold, this work I've highlighted today. I'd also like to thank collaborators, Rob Britton on the MBRAs, Sarah Blute on the organoids, Moresso on the phage, and then Indira for the microscopy work, and then funding sources. And I look forward to your questions. Thanks so much.